Let us now talk about the phases of plant growth and development. So the entire period of plant growth and development can be divided into 3 phases. And these 3 phases would be the first, germination of the seed and vegetative growth. Or we can also call it vegetative phase. The second is the reproductive phase where flowering takes place. Or flowering period when the plant reproduces. And the third phase is when the plant gets old and then dies, that is senescence and death. So in these 3 phases, the complete plant growth and development gets completed. We will be talking about all these 3 phases. Let us start with the first one, that is germination. Germination is the time when the radical emerges, the plumule emerges, and from that seed a baby plant grows. To understand what exactly happens during germination, the scientist Van Overbeek explained the complete process of this germination. The first thing that happens when the seed germinates, and when we saying it is germination, that means the dormancy period is over. So first step is imbibition. Imbibition is adsorption of water. It is not absorption, adsorption of water. Adsorption of water. So this water comes into the cell, into the seed, and gets absorbed on the surface of the cells. After this, when water gets imbibed, gibberellin secretion or concentration increases. So now there would be gibberellin increase. Third step, this gibberellic acid or gibberellin, it migrates or it is transferred or translocated to aileron layer. Aileron layer is the place where in the seed food is stored. So this is the stored place or storage where the food is reserved for the development of that baby plant or embryo. So first water goes in, it is absorbed. Then that helps or that initiates increased concentration of gibberellic acid. Now when gibberellic acid or gibberellins are more, they migrate towards the storage place. Most commonly it is the aileron protein or aileron layer where the food gets stored. So now gibberellic acid moves here. Next step, gibberellic acid initiates synthesis of enzymes. Initiates synthesis of enzymes like amylases and proteases. Amylases are the enzymes which act on carbohydrate. And proteases act on proteins. So now the reserved food would be digested with the help of these enzymes which are synthesized. And their synthesis has been initiated by gibberellic acid. Now when these food molecules are broken down, digested, now that nourishment is supplied to that embryo. So it is translocation of nutrients to radical and plumule, that is the part of the embryo. And then the first thing that happens when we say that the seed has germinated is the emergence of radicle. So when radicle emerges, that process or that emergence indicates initiation of germination. So first it's going to be emergence. Emergence means emerging radicle. When radicle comes out, that is what is known as emergence, and this will be immediately followed by plumule growth, and now the embryo would have radicle and plumule, and that is what we call the seedling or the baby plant, which indicates that now the seed has germinated. So in the first phase, germination takes place, and this baby plant which is formed is going to grow. The roots will help in anchoring the plant, absorption of water and minerals. Plumule would come out of the seed, or if it is put in the soil, and it will produce leaves, branches, and then the vegetative growth or phase starts. In vegetative growth, it is only root, stem, leaves, those structure form. And during reproductive phase, now the buds, the reproductive buds, they would produce the flowers, and then the next phase would start. So now let us talk about the conditions which are required for the germination process to take place. Let us now talk about the conditions required for germination. We just now saw all the steps which are required for germination to take place. And the first step was imbibition. And that is why the first thing which is required is water. Unless and until water is imbibed, the entire sequence of changes which takes place would not start, because it starts with imbibition of water. So water plays a very important role, and we can write here it is required for initiation of the germination process. Then the next condition which is required for germination is oxygen. Oxygen is essential because once this process begins, then aerobic respiration, aerobic breakdown of that carbohydrate which was moving or getting translocated should start, because that is what is going to provide ATP. So it is required for aerobic respiration, that is breakdown of carbohydrates aerobically. And this process helps in generation of ATP. So now this ATP would be available for all the processes which are taking place in that seed during germination. Third condition, optimum temperature. Why this temperature should be optimum? Because when we talk of the processes, we said that gibberellic acid promotes synthesis of enzymes like amylases and proteases. And all enzymes, they work best at their optimum temperature. So it is required for optimum activity of enzymes. Apart from this, other factors also play important role, like light. Now, what is the role of light in germination? There is a pigment which is known as phytochrome. Phytochrome perceives light stimulus, and it exists in two forms. There are two forms of this phytochrome. It is written as PR and PFR. Now this P stands for phytochrome and R stands for the wavelength which it absorbs. So it is phytochrome absorbing the red wavelength or red band of light. This is around 680, or we can say 660 to 680 nanometers. And PFR is the phytochrome which absorbs far red. Now, one more thing which we have to remember here is the time when these wavelengths are available. Red is available during daytime because this is a part of the visible spectrum, and far red is available during night or the dark phase. And these two forms, they are interconvertible. That means PR changes into PFR by absorbing the red wavelength, and PFR gets converted into PR by absorbing far red. That means during daytime, PR, that is phytochrome, which is absorbing the red wavelength, gets converted into PFR. So this conversion takes place during daytime, and the reverse conversion, that is PFR to PR, would take place during night time, because these wavelengths are available during those periods, and these forms are interconvertible. It has been found out that this PFR is the active pigment which promotes germination. So it is considered as the active form, and this promotes germination. And that is why exposure of light or photo period, that is the duration of light for which the seeds should be exposed to light, is essential, because unless and until a concentration of PFR is accumulated in the seed, germination would not take place. So we can say that germination is a phytochrome regulated process. Germination is phytochrome regulated process. We will be talking about one more process later on in the reproductive phase, that is flowering, and that is also regulated by phytochrome. And in case of germination, the active form of this pigment which promotes the process is PFR. So this is very important, which we have to remember. And these two forms, they are interconvertible. So when seeds get all these conditions, sufficient water which gets imbibed, then oxygen so that ATP synthesis can start, optimum temperature for the activity of all the enzymes, and light. When light is available in that precise duration, that concentration of PFR would be synthesized, and that is the active form. It is going to help in germination process. Now, after understanding the conditions and the process of germination, we now need to see the types of germination. So now we will take up the two types, that is epigeal and hypogeal germination.